to start our live event tonight. As you know, there are two events tonight, and we are starting with definitely a very, very interesting topic, which is genetic causes of infertility. And well, we have a special guest that will explain this to all of us. And as you can see, Dr. Douglas Lester is with us. And Dr. Douglas Lester is from is from Fertility Genetics, Genomics.
Fertility Genomics.
Exactly. I am very sorry, but he's here to present our topic. And of course, very, very happy to have you with us. And how are you feeling tonight? Douglas, how are you today?
All right. Oh, sorry. I thought you were. I didn't realize you were talking to me. Sorry. Yes, I'm fine. I'm fine. I'm very, very looking forward to this presentation. Sorry.
I am
was
very,
in another
very
world.
happy and excited to have you with us on board. And as always, remember that we will start with Doug's presentation, but afterwards, it will be time for your questions. So all you need to do is type those in the chat section, and Doug will answer them for you after the presentation is done. Okay, and also of course, remember that as you know, we are every single day here just because we want to support you, answer your questions, give you the opportunity to meet lots and various of fertility experts, but also coaches, psychologists. As you know, there are plenty more of those events coming up, and we are very, very proud to be able to be here every day and help you out, even if a little. And as you know, my IVF Answers is a part of European Fertility Society. Thank you, Dak, for presenting tonight. Thank you for joining our initiative here as well. And yeah, I guess we can start with your presentation, right, Dak?
Great, great. Thanks very much, Caroline. Thank you.
Thank you so much. Go ahead.
Right. Okay. Thanks. Hello, everybody, and welcome to my talk about our exciting new company, Fertility Genomics. I'm Dr. Doug Lester. I'm the Chief Scientific Officer of Fertility Genomics and co-founder of Fertility Genomics. I'd like to talk to you today about our exciting brand new company, Fertility Genomics, which aims to bring the power of next generation DNA sequencing and applying this to try to help those unfortunate couples with a genetic cause of infertility. This DNA test may be particularly beneficial for those with unexplained infertility. A positive DNA test with us can then help such people make an informed choice as to which assisted reproductive technique is best for them. Okay, I'll just go to the next slide. This slide is an attempt to summarize the current state of play in the genetics of infertility. On the left of the slide are examples of genetic conditions causing infertility that are generally picked up first by clinicians and/or fertility technicians, and may or may not be later confirmed by DNA sequencing at a later stage. For example, the conditions azoospermia, or complete lack of sperm in the ejaculate, will easily be identified down a microscope during a diagnostic semen analysis in the fertility clinic. Similarly, polycystic ovarian syndrome, or PCOS as it's here, will usually become apparent in females in their late teens or early 20s. A confirmatory DNA diagnosis is of value for those seeking to understand the cause of their condition, but does not change the treatment options to patients suffering from these well-characterized syndromic genetic infertility problems. Where fertility genomics can potentially help are the 25% of couples in the world that suffer from so-called non-syndromic or unexplained genetic infertility. Such infertile couples may continually fail conventional IVF treatment and even the direct sperm injection method known as ICSI. Alternatively, couples may be suffering from consistent embryo or implantation failure or miscarriage. It is these conditions on the right of this slide that Fertility Genomics hopes it's likely to make a positive impact. That's where we hope to help. The next slide. Yeah, so this is a slide of the strategies currently available for detecting possible genetic defects responsible for unexplained genetic infertility. Sadly, the current methods just scratch the surface. For example, looking at all the patient's chromosomes in a process called karyotyping, on the left of the slide, can be compared to trying to find a single typo error in the complete works of Shakespeare by comparing the size of books containing an individual play. Sadly, a significant difference in size is only detected by this method when there's a whole equivalent of a whole act of a play either completely missing or completely duplicated, leading to a significant difference in the number of pages. And you'd be able to look at the two books together, and you see one would be bigger than the other, or one would be smaller than the other, whatever. Such huge letter changes in your chromosomes, visibly change the chromosomes, are probably only responsible for 5% of infertility cases. They're not the most common form of DNA mutation. The other current method used is sperm fragmentation index, which I've got a Shakespeare analogy here again. It's the equivalent of looking at how well a man's complete work of Shakespeare playbooks have been stored in his library over his lifetime. If they've been stored poorly, then when you shake the books, pages that have disintegrated over time will fall out. This gives some indication that environmental factors have to some effect affected some of the sperm's chromosomes, which in turn may lead to a decreased chance of pregnancy. The validity of this test is, however, controversial, to say the least, as there are real problems with reproducibility of this assay and the fertility aspects it's correlated with. So it's not a good technique. Neither of these assays are looking at the individual letters of the DNA, or again, by analogy, the equivalent of finding individual small letter typos in the complete works of Shakespeare. And such DNA errors, small equivalent of small typos, are responsible for the vast majority, at least 85% of the causes of unexplained genetic infertility. Why is no one looking for such typos that cause unexplained infertility? The answer is because only recently has the technology been developed to be able to do this affordably. It is this paradigm-shifting technology that Fertility Genomics is now using to try to help infertile couples diagnosed with unexplained infertility. Okay, I'll just get to the next slide. Yeah, so this new technology known as whole exome sequencing is unprecedented. And it involves simultaneously sequencing all of the individual letters present in your 22,000 genes that are present on your chromosomes. This is a standardized methodology, which is currently being used for pre-implantation genetic diagnosis. It's also coming into cancer diagnosis as well. It's becoming a routine technique. Yeah, our company was started a few years ago on the back of groundbreaking research by my former colleague at Abertay University, Dr. Sean Brown, a world-leading fertility physiologist. From Dr. Brown's work, he encountered a couple, Francis and Andrew, who unfortunately failed 9 IVF cycles. From Dr. Brown's work, he concluded that the fault probably lay in one of the many genes that help control the mobility of sperm. And Andrew therefore took our fertility DNA test. Fertility Genomics' unique analysis revealed that Andrew had a 6-letter DNA deletion. There's only 4 letters in the DNA alphabet, A, C, G, and T, and Andrew was missing C, T, A, T, G, G. And you can see the normal sequence on the left, and you can see that they are missing from Andrew's DNA. Um, from these significant results, we calculated that Francis and Andrew would only have a 1% chance of fertilization by conventional IVF. From these results, Francis and Andrew were recommended to use the direct sperm injection method known as ICSI. This happily resulted in the birth of their dream baby, Heather. Following this success, we recently launched Fertility Genomics to offer our sequence analysis service to all couples in the UK. Our spit kits are sent discreetly to our customers' own home. This saliva sample is posted back to us, where we extract DNA, sequence it, and analyze the DNA for mutations in all known genes involved in fertility. The results of our DNA test will then help inform you which is the best infertility treatment to maximize your chances of conception. All of the staff at Fertility Genomics come from a university academica. Have I got the wrong? Yeah, sorry, I skipped a slide here. Let's go back. Yeah, sorry, I skipped this slide now. Yeah, but I think all of the staff at Fertility Genomics come from university academica, which allows us to continue with further research into the correlation of DNA letter changes and particular infertility problems. Part of our profits will also be plowed back into pure infertility research. Right, I'll leave that slide up just for a second. Right, I mean, this last slide is really aimed at any staff of fertility clinics who may be present this afternoon. It's a bit more technical. It tries to summarize what we do. Please feel free to contact us on any of these platforms, these digital platforms. And thank you for listening. And I'm more than happy to take any questions you may have.
Thank you so much, Dan, for that interesting presentation. What you're doing definitely sounds amazing, because I believe lots and lots of patients, well, simply would be able to use that, because that can help them solve problems a little bit earlier as well. So that's definitely something that is simply amazing. What else can I add, really? And well, now, as you mentioned, it is time for all of your questions. So if you have any questions, you know what to do. Just type those in, and Doug will try to help you out a bit with them as well. And of course, Doug, are you ready for the questions then?
Yeah, yes, I'm ready. I'm quite happy to answer questions. Yes.
Amazing. Thank you so much then. And let's have a look. Okay, we do have a very first question actually from L. Let me just go and show you. So you said that you help patients from the UK, right? So what about patients from abroad?
Yeah, at the moment we've just launched in the UK, but we're hoping to team up with DNA sequencing companies from abroad and offer it all around Europe, and America, the whole world. Yes, but initially we're just set up in the UK, so hopefully it will be available within a year. We hope to be launching to as many sites as we can in Europe. We're in discussions with a European sequencing company to enable us to move out to the whole of Europe soon.
Okay, and for how long, if I may ask, for how long you've been helping out? I mean, for how long is...
We've only launched 2 weeks ago, and we've just had our first customer. So this is absolutely hot off the press. We're just launched just now. It's taken a long time to get things into place, to be able to get all the procedures in place and everything up to clinical satisfaction. But yeah, so we've just launched now.
Okay, then I understand completely. Okay.
Yeah,
So we just need to wait until, of course, it's expanded as well abroad. But definitely
yeah.
sounds amazing. Sounds promising. Thank you so much for the very first question. And of course, let's have a look. Okay. Next question we have from Asha. So, how do you test for recurrent miscarriages, implantation failure in the female partner specifically, and what treatments are recommended based
Yeah.
on your tests?
No, this is still in a bit of a research area. This, but there are genes that definitely maybe predispose you to miscarriage. I mean, it's factor five, the Leiden variant, it's called, and that definitely predisposes you to miscarriage. Unfortunately, the treatments aren't on stream yet, but who knows if it certainly if they can come up with a treatment for that, then then a DNA finding out that you've got factor five Leiden in advance, know you're at risk of miscarriage, but they would alert the doctor in advance. And if there were new treatments available, then that might prevent the miscarriage. So this is still maybe in a research stage, but but one for the future. Implantation failure. There are definite genes that can cause failure of the embryo to implant, and yes, that would that yes, you maybe have to get almost a donor woman or something to be able to get the embryo going. But again, maybe there might be treatments available in the future for that. But these are still very experimental um, at the moment.
Hmm. Okay, thank you. And actually, Asha has a follow-up question. Okay, just give me a
Yeah.
second and let me show you. Okay, so for egg donation, do you test for, not sure here, sorry, factor?
The EGF
Okay,
factor issues in endometrium. No, that's um, we could look at that gene, the VEGF factor. This is the beauty of our sequencing company, is that because we sequence all the genes, if new research comes out and says that VEGF, a variant of VEGF, definitely causes problems, and there's a treatment, then then yes, we could easily look for VEGF. It's not on our list at the moment, but as soon as I'm finished this, I'll go and look at the information, and we can add VEGF to our list. So any genes at all, if the patient actually requests a gene, we can probably actually look at that gene. But it's only useful if there's a treatment, a positive treatment, um, would be better. But yeah, thanks for that question.
definitely sounds interesting.
Yeah,
So,
yeah.
so in such case, you are able to simply, check all possible genes then, right? So it's like it's an
Yes,
option.
yeah, yeah. So new
Mhm.
genes are coming out every day.
Yeah.
Well, not
Okay.
well, maybe not every day, but at
Hmm.
least I get updates from the main website on online Mendelian inheritance in man. Um, I get I get this I get updates from them all the time. Um, and they alert me to, oh, there's a new gene for implantation failure, there's a new gene for IVF failure, and so on, and then we just add that to our list. It's all, we're sequencing all the genes at the moment. It just means that we will actually look at that gene in the future.
All right, that's even better
Yeah,
to
yeah,
to
yeah,
to
yeah,
learn. I definitely. Okay, I'm just waiting for some questions right now, so let's give it a minute. Okay,
yeah.
let's see if we have more questions. Usually that happens very, very fast. And yeah, as I was saying, someone is typing. We need to wait a bit, but yeah, let's see what else we have here. Might take a minute.
I baffled them. Hope I wasn't too complicated. Yeah.
Those topics are never easy, that's for sure. So this is genetics, anything that, I mean, this is actually our very first webinar with you, and for us it's definitely sounds amazing that you can
Yeah.
even do that right now. I mean, of course we hear that it's getting more and more advanced, but when you hear that it's possible for you to just check some genes and see if see the what can be done with it, then definitely that's simply I'm definitely someone that maybe not that I wasn't sure. However, it sounds like next level kind of thing for sure. Thank you so much.
The
Okay.
good news is that the, good news for the parents is that usually need two bad copies of the gene before you're affected, and you will only pass on one of these bad copies to any child you eventually have. So the child itself will not suffer from infertility. I didn't actually say that in the talk, but that's that is one good news coming out of it. If we manage to identify the problem and maybe point the people towards direct sperm injection and ICSI, then if the child born from that, it's very unlikely that it will have fertility issues like the parents. So that's one good thing that comes out of it.
Definitely, that's definitely
Yeah.
true. Thank you so much. And let me show you our next question from Bubble. I may have missed it in your presentation, but
Yeah.
for problematic sperm count, how many genes would you test in a husband?
Probably at least 500.
Okay.
A lot. We will look at a lot of sperm genes. There are a lot of genes that are known to affect sperm. Azoospermia, we could look at a lot of genes for that. Um, yes, there's lots of genes for low sperm count.
Okay. All right. Thank you so much. So even over 500, you said, right? So yeah,
Yeah,
that's amazing for sure. Okay. So I think Bubble is typing follow up. So again, we just need to give it a minute and see the next question. Yeah, that's kind of my answer. Wow. So if one of these 500 is found to be abnormal, is there a therapy available? As my understanding is, we cannot change our genes, can
yeah,
we?
yeah. That's yes. It might be that we advise that IVF might not be the best route, and that direct sperm injection method in this case might be better. Um, if it's low sperm count, it might be better doing direct sperm injection method. And yet, no, we can't change our genes. But as I said before, you're only going to be passing on one of the bad genes to your child at the end.
All right. Again, thank you so much. And well, you cannot, as you mentioned, fix them, but if we know, then there are options that we can find. Yeah. And
Yeah.
that's what you are aiming here at as well, right?
Yeah. Yes. Yeah. I mean, we see a particular. Hopefully, new drugs will come on stream. This is what we're hoping that as we build up the database, as we get better and better correlations between a clinical outcome and a particular gene, then the drug companies will get interested in that particular gene. It's a drug target, and then hopefully that it might be able to sort the problem without even without IVF, like maybe intrauterine injection, or with IVF, or with a direct sperm injection method, or advanced enhanced direct sperm injection method. So yeah, we're hopeful that this is going to benefit patients, but the results of these findings, that each individual case could help further other unfortunate couples in the future as well.
All right, so that's what's amazing, definitely, because it's an option for us to learn a little bit more, and then maybe even that will make those like provoke anyone else to actually help out as well. So it's like finding out the very important but also not easy thing to do, like right genes. So thank you so much. And as you can see, Bubble has added already one of thank yous. Thank you very much. That's an exciting new area, that's for sure. Okay, we are waiting for some more questions, of course. So if you have any, go ahead and type those in. If not, of course, remember there's always an option for you to get in touch with Doug and his team, and I'm sure they will be more than happy to help you out with your questions. And let me just put the link. You can also use this link, and there will be an option for you to get in touch with Doug as well. And of course, yeah, at least at the moment, I don't see any more questions. But of course, Dag, is there anything else that you could tell us? Perhaps like how it all started. If you can share that with us, how it all started with you in this company,
Yeah.
because that's definitely amazing.
Well, it was my colleague. I was still a lecturer at Abertay, and then and fellow lecturer Dr. Sean Brown. He works with the fertility clinic, and he came to me with a with a problem patient that he had shown physiologically that the motility of the sperm was was slower than it should be, and he'd found the underlying cause of it physiologically, but it was a very laborious technique he used, and he got the patient's DNA sequenced, and he came to me to help to try and confirm this patient, to check to try actually find the DNA mutation, and it's that patient that we are presenting in the slides. It's and we called him Andrew, and he's got 6 letters of DNA missing in it, and from this, then yeah, the couple were advised for direct sperm injection method. They had a baby, and I thought, well, wow, we've helped this couple. Why we could help a lot more couples. The trouble is that there are lots of genes, there are rare variants, but we can, with this new technology, we can find them. We can still find them. It is like looking a needle in the haystack, but we can do it. And I say, well, why don't we start? Well, why don't why don't maybe I could start a company in the back of this, and I got the company off the ground, and it's taken a year or two to get the company off the ground. I've retired from academia now, and I'm with the company full time, and developed a probability score, and from the DNA we can give a probability score of conception, or not of conception, or fertilization or not. We call it a fertility probability score. But yes, it's a fortunate. My background isn't in sperm, but it's in other hereditary conditions. But I've always worked with DNA for 40 years, and I'd like to use this knowledge to help people in the infertility area.
All right, so nice to
Yeah,
hear the
yeah.
story behind it as well, because I think that it's amazing to learn that it's all started from well, it's like we are all here and our patients are here with us tonight, and they are wondering what can be done, right? And it starts right there that you need to check it, and there are other options right now as well. Thank you so much, Doug. That's definitely good to hear. And I think that it looks very, very promising, to be honest, because it's like I've mentioned, it seems like it's another level of doing things. So thank you so much for bringing this to us tonight. We will be finishing for tonight, but it's definitely something that I am very happy to discuss, and perhaps we will have more of those events coming up as well. And before I let you go, anything else you would like to add to our audience?
No, I'd like to thank Caroline and Jakob for organising this event. It's a fantastic opportunity to actually talk to be able to talk to potential patients who might be interested in doing our DNA test. But thanks very much, Caroline and Jakob.
Thank you so much indeed for also for being here with us, because as I mentioned, this is definitely interesting technology that we now can use, and I can only imagine that it's going to be more and more out there, and more patients will be able to simply check it and use that new technology as well. So thank you so much for that. And of course, thank you all for joining us tonight, for your questions. I do believe it's been interesting for you to find out about fertility genomics. So thank you so much. And as you know, we will have another webinar tonight at 6 p.m. UK time. So I also hope you will be able to join us. And once more, remember that this has been recorded, so you will have a chance to watch this again, of course. Dag, once again, thank you so much for joining us tonight. And everyone have a lovely evening and weekend. Dag, I just hope we will have a chance to have another webinar with you, and we will be able to discuss this even more, because it's definitely amazing. Thank you so much. Take
No,
care.
thanks, Caroline. Thank you. Thanks very much. Thank you,
Thank you.
everybody. Thanks.
Bye.
Bye. Bye.